What drug product is evaluated in this trial and what is its mode of action?
Our study evaluates the therapeutic cancer peptide vaccine FusionVAC22 in combination with the immune checkpoint blockade by atezolizumab. For many years, vaccinations have been used to prevent infectious diseases such as measles or influenza. The principle of these vaccinations is that part of pathogens, so-called antigens, are administered to patients. This activates and alerts the immune system to these pathogens so that in the event of a subsequent infection, they are recognized and fought by the body's own defensive system. Today, we have the possibility to use these principles also to treat malignant disease. In contrast to infectious disease, which can be prevented by vaccination, these cancer or tumor vaccines are used for those patients who have already developed cancer. So they are referred to as therapeutic cancer vaccines. The most important prerequisite to design such therapeutic cancer vaccines is the selection of the perfect antigen. In recent studies of tumor tissue from patients with fibrolaminar hepatocellular carcinoma, the so-called DNA JB1-PRKACA gene fusion was identified as a genetic hallmark of the tumor. A fusion transcript is a combined protein that is formed based on the fusion of two different genes and is significantly responsible for tumor emergence. The DNA JB1-PRKACA gene fusion is present in fibrolaminar HCC as a trigger of tumor formation. The therapeutic cancer vaccine FusionVAC22 administered in our study contains a peptide antigen derived from this DNA JB1-PRKACA gene fusion. In order for a therapeutic cancer vaccine to induce a strong and long-lasting immune response, we further need immune stimulators, as the peptides themselves are not very immunogenic. In our trial, we will apply the immune stimulator, a so-called adjuvant called XS15, which has been previously evaluated in several clinical trials and showed very good safety profile and induction of long-lasting T-cell responses in different cancer patients. Our XS15 adjuvanted peptide vaccine will be combined in this trial with an immune checkpoint inhibitor. Checkpoint inhibitors are antibodies that activate particular immune cells, the so-called T cells, by blocking the interaction of the PD-1 receptor, a marker site on immune cells, with the PD-1 ligand, which is why they are so-called checkpoint blockers. Some tumors produce the protein PD-L1 that, by binding to the PD-1 receptor, prevents the body's immune cells from recognizing and effectively fighting the tumor. By blocking the PD-1 receptor or the PD-1 ligand, immune cells are once again enabled to do their work, as the cancer is now more visible to the immune system and can be better targeted by the patient's own immune system. However, the effect of a checkpoint inhibitor is general or non-specific, which means that it's not specifically targeted against the tumor. In this particular case, the specific response of the immune system against the tumor induced by our therapeutic peptide vaccine should be enhanced and improved by the checkpoint inhibitor. The checkpoint inhibitor used in this clinical trial is atezolizumab, an antibody directed against PD-L1, which is provided by Roche.
How was the FusionVAC peptide vaccine developed?
The FusionVAC peptide vaccine, as well as the adjuvant XS15, were both developed at a University Hospital in Tübingen in the south of Germany. In extensive preclinical work, the FusionVAC peptide vaccine was characterized, and it was shown that the peptide antigens are presented naturally on the surface of tumor cells. Moreover, it was shown that these peptides are recognized by the patient's immune system, in particular by the T cells. Moreover, in first clinical application of these XS15 adjuvanted peptide vaccine in single patients with fibrolaminar HCC, we could show a good safety profile as well as the induction of strong and long-lasting T-cell responses. More important, these T-cell responses were accompanied in these single patients by long-lasting disease-free survival, which really gave us hope to establish this clinical trial.
What are the goals for this clinical trial?
A major goal of this study is to verify whether an immune response can be generated by the FusionVAC XS15 vaccine in combination with immune checkpoint inhibition with atezolizumab. This is a prerequisite for the immune system to fight the tumor. Similarly, the tolerability and toxicity of the FusionVAC XS15 vaccine in combination with immune checkpoint blocker therapy will be assessed. Moreover, we will assess in all patients the clinical response to the combination of peptide vaccination with immune checkpoint inhibition.
Who can participate in the clinical trial?
In our study, patients can be included with a locally advanced or metastatic fibrolaminar HCC. Moreover, patients with other malignancies that are locally advanced or metastatic and show evidence of the DNA JV1 PRKACA fusion transcript can be included in the clinical trial.
How does the trial schedule look like?
When patients are registered for the clinical trial, they at first are included in a pre-selection process. The pre-selection process has the purpose to determine if a patient can participate in the study. This pre-testing usually takes about 3 to 4 hours. Only when the results of all tests are available, the investigator will be able to tell whether the patient is eligible for the study or not. After the screening, the vaccination and therapy phase will start. In this phase, the patient will receive at least 2 vaccinations with the FusionVAC XS15 peptide vaccines as part of the study. The vaccinations are given subcutaneously, which means that the vaccine is injected under the skin of the abdomen with a small needle. To study the effect of the vaccine on the immune system of the patients, blood samples will be taken periodically. There's a possibility of a revaccination after month 11 if the immune system of the patient to the initial 2 vaccinations are already measurably decreased by that time. Beside the peptide vaccine application, the patient will receive the checkpoint inhibitor atezolizumab as intravenous infusion every 4 weeks. The treatment will be given for a total of 12 months unless there's a sign of disease progression or other reasons to discontinue the study. After completion of study treatment and vaccination, the patient will enter the follow-up phase after the last atezolizumab infusion. There is the necessity for the patient to come to the hospital for 4 more times to check for follow-up visits. The last checkup and end of the study is 6 months after the last treatment. The FusionVAC22 trial is recruiting since October 2023 at the University Hospital in Tübingen in the south of Germany. We are currently preparing to open several other centers in Germany, and this trial is also open for any international patient. If there are further questions from the patients or their physician, please feel free to contact us and we will be very happy to answer all your questions.